Well, we've covered the categories of carbohydrates, so this next part of the lecture deals with carbohydrate transports. We're going to talk about mechanisms and patterns of carbohydrate transport. A concept that's important for understanding and thinking about the transport of carbohydrates are sources and sinks. Sources are just places from which carbohydrates are exported. Probably the most common or obvious source for carbohydrates is leaves. Leaves do photosynthesis and fix carbohydrates and then export those carbohydrates elsewhere where they're used. But also, sources can be places where carbohydrates are stored. So for instance, in a deciduous tree that's leafing out in the spring, the carbohydrates for that initial part of leaf expansion come from places where carbohydrates are stored. So in that case, the sources are branches, stem tissue, or even roots where carbohydrates are stored. So those are examples of sources. Sinks are just locations or tissues where carbohydrates are used. So in reality, any living cell is a sink for carbohydrate because they use carbohydrate just for maintenance. In reality, the strongest sinks are rapidly growing meristems, places where growth is occurring rapidly. So the vascular cambium or the apical meristem is a strong sink for carbohydrates. But also phenologically at certain times of the year, ray cells in bark, stem, or roots may become strong sinks for carbohydrates because carbohydrates are being stored there. So that could also be a sink. So I've mentioned a couple of times the idea of sink strength. Sink strength is just a qualitative attribute that refers to the magnitude of carbohydrates that are being used in a particular location, but also the distance from the particular source. So for instance, we may speak of the apical meristem as being a strong sink for carbohydrates relative to upper leaves, for instance. And that's a function of both the amount of carbohydrates being used, but how far away they are from any particular source as well. In general, as a rule of thumb, for instance, the leaves in the upper canopy tend to supply most of the carbohydrates for rapidly growing apical meristems. And that's just because they're closer. And that's the strongest sink. Whereas leaves in the lower canopy tend to supply more of the carbohydrates for sinks in the stem and roots. And that's just a function of magnitude and distance. You can really think about sinks as basically being a pull on carbohydrates. So those strong sinks tend to pull carbohydrates towards them from the closest sources. So that's one way to think about sources and sinks in plants. One thing to think about is how rapidly do carbohydrates move in the phloem. Here's some data that looked at a number of angiosperm and gymnosperm species. And what you can see is that angiosperms in this review, maybe had transport velocities around 80 to 100 centimeters per hour. So maybe a maximum transport velocity of around a meter per hour. Whereas gymnosperms tended to have slower transport velocities in their phloem, maybe 20 or 30 centimeters per hour. So thinking about that relative to maybe a day, angiosperms, maybe in a day you have 24 meters of transport, whereas in gymnosperms, you might have less than 10 meters of transport through the phloem in a day. So this is relatively slow transport, and trees are tall. So when we think about it, for instance, in a 100-meter tall gymnosperm or a redwood, it might take 10 days for a molecule of sucrose to move from the crown down into the roots. So we're really talking about relatively slow transport here. Transport in the xylem is a little bit faster. We do know that this transport is not diffusion. Diffusion is really slow. If transport of carbohydrates relied on diffusion, it would take years for molecules of carbohydrates to move from the crown to the roots. We know that there's some other process that is responsible for phloem transport. And transport molecules, we've already talked about how most sugar transport occurs in oligosaccharides like sucrose. We talked about that in the previous part of the lecture. All right, so what is the mechanism then? We know that diffusion is not how sugars move in the phloem. So what is the mechanism of phloem transport? The mechanism of phloem transport is sometimes referred to as the Munk hypothesis, or the Munk pressure flow hypothesis. So Munk pressure flow, basically you can think about this as a series of events or steps that happen. And the first is sugars are actively loaded into the phloem at the source. Okay, so there's some active process, and we'll talk about phloem loading in a minute. There's some active energy requiring, when I say active, I mean energy requiring process by which sugars are loaded into the phloem at the source. So that's active. That process, that increase in concentration of sugars in the phloem, lowers the osmotic potential of the phloem at the source, and that causes water to flow into the phloem. Okay, so that's what we're seeing here. This is water flowing into the phloem after the active loading of sugars into the phloem. So that increase in concentration of sugars decreases the osmotic potential, and we'll talk more about water potentials in the water relations part of the course. But suffice it to say that lowered osmotic potential causes water to flow into the sieve tube elements at the source. Okay, so that basically pressurizes the phloem at the source. So you can sort of think about that as sieve tube elements at the source are pressurized. Okay. Meanwhile, at the sink, so at the destination, sugars are unloaded And this may be passive or active, depending on the situation and the biology of that particular species. So at the sink, those sugars are unloaded. So those sugars are taken out of the phloem. So of course, that causes the osmotic potential of the sieve tube elements at the sink to rise, and that causes water to flows out of sieve tube element. Okay, so at this point, water flows out of the sieve tube element, and that lowers the pressure. So that means there's a pressure differential from the source to the sink. So sugar flow in the phloem, I'll just say follows the pressure gradient. It's like a garden hose. So basically the loading of sugars at the source pressurizes the phloem, and that causes then, and then the unloading of sugars at the sink depressurizes the phloem, and that causes a pressure differential. And so phloem transport is down that pressure differential. So that whole process is referred to as Munk pressure flow. So how do the sugars get loaded into the phloem? There's two main mechanisms or types of loading that occur. And just for clarification, these diagrams that we're looking at, this is a cross-section of a leaf. What they're showing in this blown up portion are spongy mesophyll cells where most of the photosynthesis occurs, and their connections with each other, and then subsequent connections and transport of sugars to the leaf vein. So that's, where the nearest phloem cells are, is in the leaf veins. So that connection is sort of happening right here. So this is in the vein right here, and these other cells are the mesophyll cells where photosynthesis is occurring. And there's two main types of loading: symplastic and apoplastic. So symplastic just means functions or things that happen within cell membranes. So it usually means doesn't involve passage across a cell membrane or across a cell wall. So symplastic loading means the loading occurs through plasmodesmata connecting multiple cells without crossing a cell membrane and cell wall, whereas apoplastic loading means at some point the carbohydrates leave a cell and has to cross cell membranes and cell walls. Right now, this is still an active area of research in terms of exactly which species of plants do which kinds. There's evidence out there that some tree species do symplastic loading, while most herbaceous plants do apoplastic loading. But like I said, it's still, I think, up in the air to some extent. So really, when we're talking about trees, most of the evidence right now suggests that they use symplastic loading. And in this diagram, you can see with symplastic loading, the passage from mesophyll cells to the phloem sieve tube element is through plasmodesmata. So you can see these gaps between cells. That's where there's basically a pore in the cell wall, and the cell membrane is continuous between all of those cells. Now, the loading into the sieve tube elements is still active. And in this case, probably what that involves is the active transformation of monosaccharides that are transported through the mesophyll cells into the intermediary and sieve tube elements, the transformation of those monosaccharides into oligosaccharides, either sucrose or even bigger oligosaccharides. And what that does is it prevents those are now bigger molecules. It prevents them from diffusing back outwards back into the mesophyll cells. On the other hand, with apoplastic loading, it probably involves proteins in the cell membranes of these companion cells and involves the active transport of mono and maybe oligosaccharides across the cell membrane into the companion cell and sieve tube element of the phloem cells at that point. But in both cases, that's an active, energy requiring step. Now, I already mentioned that at the sink, there's unloading of sugars from the phloem. And the evidence shows that that unloading step may be either active or passive. So in some cases, that may just involve the diffusion of sugars from the phloem, an area of high concentration, towards the sink, an area of low concentration of sugars, and that would be passive. Or it may again be an active unloading of the phloem at that stage at the sink as well. So it may either be active or passive at the unloading step. This next bullet about information transmission via phloem pressure waves is a hypothesis that Missy Holbrook and her group up at Harvard put forward to deal with a problem, especially in trees. So we know that in plants, there's information transmission in terms of coordination of sources and sinks. And in some cases, those messages get transmitted relatively quickly. But for instance, in trees especially, we know that phloem transport's really slow. And it may take days for instance, increased sugars loaded in at the source in the crown reach the roots. But there's data that suggests that information transmission happens quicker. And Missy Holbrook and colleagues suggested that it's actually the propagation of that water pressure associated with loading of the phloem that happens more quickly in the phloem, and that may then transmit information, and that may be the mechanism by which those messages are transmitted from source to sink, for instance. So what they're showing here is steady state, and this is from source on the left to sink on the right. And what they're showing is that here's an initial steady state, and at this point there is increased loading of solutes into the phloem, and that's associated with increased loading of water. And that's associated with the propagation of a pressure wave, and that shading, that increased shading, you can see that travels faster than the little circles, which are the solutes. So she's showing that rapid propagation of that pressure wave down the phloem actually allows information to be transmitted and perhaps sends messages about, for instance, increased availability of carbohydrates at the source to sink locations. And that may be a mechanism of information transmission. Again, this is a hypothesis and an area of ongoing research, but it's a plausible mechanism for how information may be transmitted in a situation where the actual sugar transport is relatively slow. When we think about leaf development, there's a transition from the leaf being a sink for carbohydrates to the leaf being a source. So we can think of that as the sink-source transition of leaves. So this is an image of an expanding silver maple leaf. This is maybe a few days after bud burst, and this leaf right now is rapidly growing. There's a little bit of photosynthesis occurring in that leaf, but certainly that leaf is still consuming more carbohydrates than it's producing. So this leaf at this stage of development is still a sink for carbohydrates. At some point, that balance will occur where that leaf is fully expanded, it's not growing anymore, and it's photosynthesizing at a rapid rate. And at that point, it makes a transition from being a sink for carbohydrate to being a source. There's some data out there that there are some constraints on carbohydrate import in fully developed leaves. Whether that's physiological or mechanical is not clear, but there's some evidence that once they're fully expanded, leaves just don't import carbohydrate anymore. That transition from being a sink to being a source tends to happen more rapidly in deciduous trees than in evergreen trees. Deciduous trees, their leaves become a source more rapidly than evergreen leaves. And that's not surprising. Evergreen leaves, just in terms of being evolutionary advantageous, they would really need to become sources much more rapidly because their lifespan is relatively short. So in order for it to be beneficial to invest carbohydrate in their development, they would need to become a sink relatively quick in order to export carbon to the rest of the plant. So that sink source transition happens more rapidly in deciduous trees than in evergreen species. There's a concept called branch autonomy that was developed in the 90s, and at the time it centered around this concept that trees are really a series of repeating units. And one question is, to what extent is a branch on a tree an autonomous unit for carbon? Does that branch import carbon from the rest of the tree? Or once it becomes a source of carbohydrates, is it sort of on its own? It can't import carbon. And there was a lot of research on this in the 90s. Some of that research was centered around a time when people were using branch chambers to, for instance, do studies on ozone exposure or elevated CO2, where they would place chambers around branches and say, okay, if we expose this branch, can we infer the responses of this branch and extend that to how a tree might respond to that treatment? So this branch autonomy thinking and experimentation was was related to that idea. But I think it's also a useful thing to think about in terms of thinking about how branches behave relative to the integrated tree that they're attached to. And there's some data out there that suggests that branches are relatively autonomous with respect to carbon. And some of those sources of data are labeling experiments. So those labeling experiments suggest that after branches become a source, so after they become a net exporter of carbohydrates. They do not import carbohydrate from the rest of the tree. So these are situations where researchers have used like radioactive or stable carbon isotopes to label carbohydrate in the tree and then follow that carbohydrate around. And what they find is that in general, branches don't import much carbohydrate. Other sources of studies are pruning studies. So this picture shows someone pruning a tree. And those pruning studies show that when the lower branches of trees are pruned, that almost never has an effect on the rest of the tree. So if, for instance, the lower branches in the shade were importing carbon from the rest of the tree, you might expect, for instance, if that was happening, you might expect pruning to rapidly accelerate tree growth, and that doesn't happen. But also pruning the lower branches, especially like the lower third of branches, doesn't generally harm the growth of the trees. So those lower branches, it appears, are sort of at a net neutral state where those shaded branches are at sort of a break-even point. They're probably fixing enough carbon to maintain themselves, but maybe at that point they're not exporting much carbon to the rest of the tree. Also, when people have counted rings, growth rings, in lower branches on trees, they often see dropped rings, indicating that there's not much growth in those lower branches, which also suggests they're not importing carbohydrate from the rest of the tree to support growth of those branches. And the third line of evidence that branches are relatively autonomous is from fruit set studies. When scientists have defoliated branches on fruit-bearing trees, so for instance, removing the leaves from a branch, they find that fruit set and fruit development declines. What that means is that there's not a lot of carbon imported to that branch from elsewhere into the tree to support fruit development. So most or all of the carbohydrate supporting fruit development is coming from the leaves on that branch, which means that branch is relatively autonomous, at least with respect to carbon. So there's a few take-homes from the branch autonomy literature. The first is that old shaded branches are on their own. So that means those lower branches in canopies are not going to import carbohydrate from the rest of the tree. And that also then indirectly implies that probably one of the mechanisms of branch death, when we see canopy recession, that one of the mechanisms is just carbon starvation of branches in the lower canopy. The other take home is under normal conditions, most branches are relatively autonomous during the growing season. So most branches are producing at least enough carbohydrate for themselves, and if they're higher in the canopy, for export to the rest of the tree, and that the exceptions to autonomy that occur are during periods of strong sinks in branches. So rapid foliage production in a branch or fruit set and rapid fruit growth may cause some carbohydrate to be imported from elsewhere in the tree. But as we saw with fruit set studies and such, it's probably still minimal. So for the most part, branch autonomy appears to hold, and we can think of trees in that way. I wanted to show a couple of examples of data that sort of touch on the idea of branch autonomy as it relates to loss of branches, for instance. So I mentioned that branches in the lower canopy, because they are autonomous and generally don't import carbon from the rest of the tree, that's one potential mechanism of branch loss and the subsequent recession of crowns in tight stands. This is an example from Scots pine, Pinus sylvestris, and they measured the carbon balance of 6 lower canopy branches over time in these Scots pine. And this is interesting and sort of granular data, and I think it's a neat demonstration of some of the concepts we're talking about. So what this graph shows is the bars are, I believe, daily carbon balance. So the positive bars show the amount of carbon fixation in any particular day, and then also the respiratory cost for that particular day. So if the positive bar is larger than the negative bar, then there's a net positive amount of carbon produced by that branch. So you can see, fluctuations. For instance, this was probably a really cloudy or rainy day, so there was almost no carbon fixation, but there are always respiratory costs associated with that. And you can actually see during that sort of shady period, there's a dip in this line, and this line here is cumulative. So this dark line is basically the cumulative sum of those positive and negatives. So you can see over this period of time, the positive bars are generally larger than the negative bars, so there's an accumulation of carbon produced by that branch, and presumably that's being exported to the rest of the tree. But then at some point here in late summer, you can see those positive bars become smaller and do not outweigh the negative carbon costs associated with that branch, and eventually the carbon balance of that branch becomes quite negative. And that period when that branch carbon balance becomes negative is associated with here, this bar, which shows the period when the branch started senescing. So that's the point where they started to see needle death and eventually complete senescence of that branch. So this is nice data, carbon balance data, sort of linking the carbon balance of those branches to their eventual senescence. This next set of data is in Abies amabilis, Pacific silver fir, and this was taken in a really dense stand of Pacific silver fir. This is a photo of that stand. This is in the Cascades in Washington State. Really dense stand, trees close together, and also wide distribution of tree size in the stand. So we're looking at, dominance and codominance in the stand right here, but there also are some small suppressed trees in the understory. And this species is really shade tolerant. So small trees in the understory can persist in low light, sometimes for decades. So that's just a bit of context. So what these data show is they went in and sampled these trees, and you can see this is height of the trees, and then they ordered those trees by height. So the trees ranged from 0.5 meter in height to about 5 meters in height. And then on some of the trees, they measured where new foliage was being produced. These trees retain 6 or 8 years worth of foliage on them. But this graph, these bars show where new foliage was being produced. And then there's another panel here that shows also with height how open the canopy is. You can think of this as light, where 1 is full sunlight and 0 is completely dark, almost completely dark. And what you can see is that going down in height, obviously it gets darker. So at this level in the canopy, it's very dark. These are dense stands, really high leaf area index. So a couple of things to observe is that we do see crown recession in these taller trees. You can see that they really don't have foliage, below about 2 meters, and that they're only producing new foliage on the upper part of the crown. So they're only producing foliage where it's brighter. Interestingly, on these smaller suppressed trees, they have foliage, at light levels that are below where the big trees even have foliage. So, and that's because they can't produce foliage any higher. So the foliage on these trees is likely quite acclimated to low light levels and exists at very dark light. So, one thing this points out is that these trade-offs related to, for instance, the senescence of branches, to some extent depends on the environmental conditions in other parts of the tree. These trees, it's advantageous to continue producing new foliage in the light, whereas that's really not an option for these other trees. So they have to skin that carbon balance cat in a different way, probably by producing foliage that's acclimated to those very low light levels. So really neat, I think, example showing the interplay between light and the within individual trade-offs that exist with regard to carbon balance and the allocation of resources.